As stated in Part 1, a common goal is a healthy, long life. To obtain that, we typically read on the subject and watch videos, among other things. Something we run into quite quickly is contradictory information, which leads us to look for corroborating and refuting evidence. Let's continue covering a broad outline of useful health and longevity research parameters to help with that and other aspects of interpreting scientific claims. Welcome back to ForGevity, where we focus on topics in health span and lifespan applicable to humans. Coverage spans practical nutrition, supplementation, physical and mental fitness, sleep, and other topics, along with empirical strengths and weaknesses of various protocols for all types of people, sex, age, condition, etc. In Part 1 of this series on health and longevity research parameters, we laid the foundation for navigating the complexities of health and longevity research. We examined the pitfalls of relying on headlines. We outlined how to critically assess study quality, asking whether an intervention improves all-cause mortality or merely changes one cause while worsening another. We also discussed the importance of health span and how evolving views of aging as a disease may shift the way studies are framed. Finally, we began breaking down the hierarchy of evidence, from anecdotes to interventional studies, setting the stage for deeper dives in the rest of the series. Now in Part 2, we'll pick up where we left off by explaining model organisms and then continue breaking down the hierarchy of evidence from observational studies through peer-reviewed, randomized, double-blind, placebo-controlled trials, an additional type of interventional study. Model organism studies. Definition: research conducted in non-humans to assess physiological, behavioral, or disease-related outcomes. Why it matters: provide in vivo context and safety data before human trials. But importantly, species-specific differences can limit translatability. Species-specific differences. The more evolutionary distance between the model organism and humans, the less reliable the results may be for predicting human outcomes. Rodents, for example, metabolize drugs differently and may exhibit immune or neurological responses that do not mirror those of humans. This is why findings in mice often fail to translate to human trials. It underscores the importance of eventually validating results in human studies despite promising animal data. Typical model organisms. Yeast. Simple unicellular organisms often used to study fundamental biological processes like cell division, metabolism, and aging. Their short generation time and ease of genetic manipulation make them ideal for mechanistic studies of longevity pathways like TOR and sirtuins. C. elegans. A microscopic roundworm, Habditis elegans, commonly used in aging research. Despite its simplicity, it shares many genetic pathways with humans and has a fully mapped nervous system. Its short lifespan, 2 to 3 weeks, allows rapid testing of interventions. Fruit flies, Drosophila melanogaster. A classic genetic model with extensive tools for manipulating gene expression, they are useful for studying neurodegeneration, circadian rhythms, and gene-environment interactions. Lifespan interventions can be tested within 1 to 2 months. Zebrafish. Transparent embryos and rapid development make zebrafish valuable for studying vertebrate development, regeneration, and toxicology. Their genetics are closer to mammals than flies or worms, and they are increasingly used in drug screening. Mice. The most widely used mammalian model due to their genetic similarity to humans and availability of disease models. Lifespan studies can take 2 to 3 years. Genetically engineered strains allow for studying specific genes' roles in aging and disease. Rats. Larger than mice and often preferred for behavioral, cognitive, and pharmacological studies. Their physiology is well characterized, and they're commonly used in cardiovascular and metabolic disease research. Dogs share many age-related diseases with humans, including cognitive decline and osteoarthritis. Pet dogs in longitudinal studies help bridge the gap between lab models and human trials. Aging is natural, not induced. Monkeys. closest to humans genetically and physiologically, making them powerful models for studying complex diseases and aging. Ethical constraints, cost, and long lifespans limit their use. However, they provide the most translatable preclinical data for interventions targeting aging. Emerging model organisms. Killifish. A short-lived vertebrate with a natural lifespan of 4 to 12 months, the African turquoise killifish enables rapid testing of aging interventions in a vertebrate context. Its short life and genetic tractability have made it an attractive new model for studying lifespan and age-related decline, especially in relation to vertebrate-specific physiology and tissue regeneration. Naked mole rats. These eusocial rodents live unusually long for their size, up to 30 years, and exhibit remarkable resistance to cancer, neurodegeneration, and pain. They remain fertile late in life and show little age-related decline in function. Their unique biology makes them an emerging model for studying mechanisms of healthy aging and disease resistance in mammals. Limitations of human trials due to lifespan and ethics. Human trials may be infeasible when studying long-term outcomes like cancer or neurodegeneration because humans live much longer than common lab animals. Observing these outcomes would require decades. Moreover, ethical standards prevent assigning people to harmful exposures or denying them known effective treatments, limiting the use of randomized controls in many cases. Epidemiological studies. Epidemiology is the study of the distribution and determinants of health-related states or events, including disease, and the application of this study to the control of health problems. In simpler terms, it's the science of understanding how diseases and other health conditions spread within populations, who is affected, and why. Epidemiologists use a variety of study designs that range from observing natural patterns to actively testing interventions. These approaches fall into three broad categories. 1. Descriptive studies, which track patterns across time, location, and demographics. 2. Analytic studies, which probe deeper into suspected links or hypotheses. And 3. Experimental studies, such as clinical or community trials. In observational designs, researchers monitor health outcomes without interference, while in experimental designs, they deliberately introduce or withhold factors to assess their effects. Observational studies. Observe what happens or happen without attempting to control anything. Case reports. Definition. A detailed report of the symptoms, diagnosis, treatment, and follow-up of a single patient. Case reports typically describe an unusual or novel occurrence. Why it matters. Case reports are the first line of evidence when new or rare phenomena are observed. They can signal early warnings about adverse effects or lead to new hypotheses, though they are limited by lack of control and generalizability. Case series. Definition. A group of case reports that involve patients given similar treatment or exhibiting similar symptoms or outcomes. Why it matters. Case series can reveal patterns and generate hypotheses for future, more rigorous research. However, they lack control groups and cannot establish causality. They are valuable for identifying rare events, emerging conditions, or unexpected side effects. Case control studies. Quote, a case control study is a type of observational study in which two existing groups differing in outcome are identified and compared on the basis of some supposed causal attribute. Close quote, Wikipedia, N&D 1. Ecological studies. Quote, most epidemiological investigations of etiology are observational. They look for associations between the occurrence of disease and exposure to known or suspected causes. In ecological studies, the unit of observation is the population or community. Disease rates and exposures are measured in each of a series of populations, and their relation is examined. Often the information about disease and exposure is abstracted from published statistics and therefore does not require expensive or time-consuming data collection. The populations compared may be defined in various ways. Close quote, BMJ, ND, 2. Cohort studies, observational or interventional. Definition. Studies following a group sharing a characteristic, for example, age, behavior, exposure, over time to compare outcomes between exposed and unexposed individuals. Why it matters. Cohort studies track how different exposures relate to outcomes over time. Observational cohort studies do not involve any researcher-assigned interventions. Instead, they monitor natural variations in behavior, lifestyle, or environment, for example, smokers versus non-smokers, and assess associated risks. Interventional cohort studies, also called non-randomized interventional trials, assign treatments or exposures without using randomization. These can suggest causality, but are more prone to selection bias than randomized trials. Both types can reveal incidence and temporal relationships, but they require large samples and long follow-up to be informative. Peer-reviewed, randomized, double-blind, placebo-controlled trials. Interventional. Peer-reviewed. Definition. Research vetted by independent experts before publication, ensuring methodological rigor and ethical standards. Why it matters. Peer review filters out flawed work and enhances credibility. However, bias and variability exist across journals. Elsevier, ND, 3. Randomized. Definition. Participants are assigned to intervention or control groups using a method based on chance, randomization, rather than personal choice or doctor discretion. Why it matters. Randomization minimizes selection bias and ensures comparable groups. This helps isolate the effect of the intervention and supports causal inference. Without randomization, differences between groups could account for differences in outcomes. Suresh, 2011. 4. Double-blind. Definition. Neither the participants nor the researchers know who is receiving the treatment or the placebo during the study. Why it matters. Double-blinding reduces bias in both reporting and measuring outcomes. Participants may report better outcomes if they believe they are getting the treatment, and researchers might unconsciously interpret data differently. Double-blind studies increase reliability and objectivity. Armitage et al., 2002. 5. Placebo-controlled. Definition. A placebo is an inactive substance designed to look and feel like the real treatment, but has no therapeutic effect. Placebo-controlled means one group receives the actual intervention while the other receives a placebo. Why it matters. Placebo controls help determine whether observed effects are due to the treatment itself or other factors like expectation or natural course of the condition. This allows for a clearer measure of the treatment's true effect. Hurubjartson and Goethe, 2001. 6. Combined impact. Together, peer-reviewed, randomized, double-blind, placebo-controlled trials are considered the highest standard in clinical research. They minimize bias, control for confounders, explained in our next video of this series on health and longevity research parameters, and support causal conclusions rather than mere correlations, making their findings foundational for clinical guidelines and regulatory decisions. Please pause the video to like, share on social media, subscribe, turn on notifications, see the video's bibliography and links in the video's description, leave a thoughtful comment, and engage with others in the comment section. Then, unpause the video for any further information. Thank you. For this channel, one size does not fit all. In addition, civility between dietary camps or tribes, vegans, carnivores, and others, is encouraged. ForGevity deliberately avoids a know-it-all approach. Settled science often becomes unsettled upon new research. ForGevity may further the process by raising new questions for researchers and scientists to address. That concludes Part 2. Join us for Part 3, where we'll continue research parameters and pick up by explaining confounders. Disclaimer. No medicine is practiced by ForGevity. Materials provided are for educational purposes only and are not intended for medical advice, diagnosis, or treatment. Please consult a licensed medical practitioner. Credits. Research, script, and audio-video development by Tom Usher. Copyright 2025 by ForGevity. All rights reserved.